Next, we're going to hear about the fascinating research highlighting an innovative approach to heart cell research. Moderating the panel will be Dr. Donna Roberts, Deputy Chief Scientist at the ISS National Lab. Donna recently joined the ISS National Lab, where she will assist the Chief Scientist in developing long-term microgravity research goals. Prior to joining CASIS, Dr. Roberts was a professor in the Department of Radiology and Radiological Science at the Medical University of South Carolina. In addition to clinical work as a practicing neuroradiologist, she served as a principal investigator for several NASA-funded studies investigating the impact of space flight on the human brain during long stays in microgravity. Join me in welcoming to the stage Dr. Donna Roberts along with her guests.
Good afternoon, everyone. Is everyone having a great time here in Seattle? Learning a lot, making a lot of connections for sure. I want to thank the programmers for having us here and having this session. I think they did an amazing job with this whole conference. I do want to say, though, that they made one mistake. And in the program, they called me Dr. Roberts, the chief scientist. And that's actually Dr. Michael Roberts, my father, so just kidding. I'm very fortunate here to have a couple guests with me. Today we're going to be talking about a really interesting topic, and that is how to actually get down to doing the research that makes use of the International Space Station and brings these benefits back to Earth. And the topic today is going to be on heart cells. And so we have a very experienced investigator here. This is Dr. Sharma. He is a seasoned space station researcher. He started his space station research career, completing his PhD at Stanford University, working along with Dr. Joseph Wu at the Stanford Cardiovascular Institute, where they launched beating heart cells to the orbiting lab in 2016. After that, Dr. Sharma, in conjunction with research partners at Cedars-Sinai in Los Angeles, have launched investigations on both Axiom Space private astronaut missions, which I'm excited to hear more about in this Fireside chat. So thank you for joining me, Dr. Sharma.
Thank you so much.
I'd also like to introduce Stephanie Countryman from BioServe Space Technologies, and just as a little aside, BioServe Space Technologies in Boulder, Colorado is where I actually got my start in the space field many years ago. I won't say what year, but Stephanie has been with BioServe for more than 20 years as the company is focused on building, prepping, engineering, and ultimately executing life sciences investigations on the space station. Stephanie has been involved with supporting more than 50 different life sciences investigations, and the BioServe team has been a valued implementation partner to the ISS National Lab community, providing guidance and insight to seasoned research teams and those new to the space science. So we thank you very much. Thank you. So why don't we get started? I will turn to you, Dr. Sharma. Why don't you tell us a little bit about why you do the research that you do?
Sure, I'd be happy to talk a little bit about the work that we do. On the broadest level, of course, there's been quite a large amount of literature that's been accumulated over the last many decades about how the human body functions in space, and we've been talking about that here at this particular conference and have been talking for a while now. So at the heart, I'm a cardiovascular biologist. I study the heart and what goes wrong with the heart in my laboratory. At the cardiac level, at the heart level, there's an understanding that the heart changes shape and size and function in low gravity and in space flight. Again, this has been documented for years with astronaut studies for many years, but there's less of an understanding of what actually happens at the level of the single cell. The individual cell that comprises the human heart, so the billions of these cells, are called cardiomyocytes, and these are beating heart muscle cells. Now, we don't have as much of an understanding of what happens to those individual heart muscle cells in space. And so what my research group at Stanford did back a few years ago is we sent a sample of stem cell-derived heart cells to the International Space Station. This is a project supported by BioServe and launched in 2016. We wanted to understand exactly what's happening to the individual cells of the heart cells of the human body in space. And to do this, there was no biopsy, open heart surgery, anything like that involved. We were using a very cutting-edge technology in stem cell biology to mass produce these human heart cells in a dish. These are called induced pluripotent stem cells, or cells that you can just derive from a small sample of someone's own skin or blood, in a process that takes a few weeks to complete. And once we're able to make the beating heart cells, we sent them to the space station for a period of 1 month, and we wanted to examine what was going to change, what's going to change with these cells over extended spaceflight. And we published our results a couple years ago, and we can talk more about what we found. But basically, we saw at the cellular level, the heart is able to compensate for the reduction in gravity in the similar way to what happens to the whole heart. So the cells of the heart behave similarly to the heart as a whole. And this is a really exciting study because it helps us understand exactly what's happening to the individual cells of the human body in space.
Okay, thank you. So having done this type of research for a while now at the human level, when astronauts go into space and they spend long time in space, like as you mentioned, issues happen to the heart. There's cardiac atrophy that happens over time. There's increased stroke volume, decreased heart rate or variable heart rate, decreased blood pressure, and sometimes arrhythmias. So how does the results that you're finding at the cellular level relate to some of the findings that we're seeing in the astronauts?
Right, so what we were able to see at the cellular level was what we call a compensatory effect. So the cells are able to compensate for the reduction in gravity by changing their genetics, by changing their function in terms of how they contract. We saw some changes that were happening at the cellular level in terms of the contractility. These cells were actually beating less in space. One of the things that we saw that was very exciting was a lot of these changes reverted back to normal when the cells actually returned back to the planet. And again, thanks to BioServe for the assist with this, we were able to bring live cultures of these cells back from the space station to our laboratory at Stanford University and further grow these cells and further analyze them. And so what we saw in some ways parallels what happens to astronauts as well. You see these forms of atrophy at the muscular level, at the skeletal level, but for the most part, these changes revert back to normal when the individuals return back to the planet. And we saw something, again, very similar at the cellular level as well. So it gives you an idea of just how plastic, how adaptable the individual cells of the human body are to the rigors of spaceflight.
And can this information that you're learning be translated back to benefits here on Earth?
Certainly. One thing that we're really interested in developing are potential countermeasures. So perhaps we can utilize these cells to develop countermeasures for muscle atrophy and cardiac atrophy that could happen as a result of extended spaceflight. But a lot of these changes that occur to the cells, these are observations and changes that can also be applied to diseases of the heart that happen here on Earth. There's diseases called cardiomyopathy, damages to the heart, muscle abnormalities found in the heart. And in some ways, the phenotypes and what we see with these diseases on Earth parallels with what's happening to hearts in space as well. So one thought is if we discover something, whether it's a biologic, a target, we can use that application for both ground side studies and space studies as well.
I think one of the aspects that we're learning about spaceflight is it has this property of accelerating aging, and perhaps similarly with heart cells.
Yes.
same processes?
Absolutely, yeah. I mean, that's definitely an area of interest is accelerated aging because as we've discussed, here at the conference, that is something that, folks experience when they spend extended time in space. And we can model these forms of accelerated aging using the cells that we fly to the International Space Station because certainly there are diseases of accelerated aging that are happening on the ground, and unfortunately we're all getting older as it is. So we'd like to just get a better understanding of how we can replicate some of those aging processes using the amazing environment and laboratory that is the space station.
Okay, great. Well, we can talk a little bit more about the science in a second, but what I'm really interested in is as a researcher, it's always exciting to imagine doing your science in space, but there's this appearance of a high barrier to get into that. So I want to understand what got you into doing science in space and what helped you overcome that barrier, and then kind of segue into your work at BioServe and how you help investigators overcome that barrier.
Sure. I was very fortunate to be raised in an environment that was always very pro-space. I grew up in a city in Alabama called Huntsville, and I'm sure a number of you are very familiar with Huntsville, home of Space Camp and Marshall Space Flight Center. So it's something I've always wanted to get involved with. Of course, that makes perfect sense as to why I decided to become a biologist, right? But somehow during graduate school, I managed to intertwine those two passions through this project that we launched in 2016. And it's something that I've stuck with ever since. I have my own laboratory in Los Angeles now at the Cedars-Sinai Medical Center, and we're pursuing further experiments and studies aboard the International Space Station. In fact, one that just came back just about a month ago, or was completed about a month ago. And so, for me personally, it's something that I've always dreamed of being involved with, but I also think these days it's easier than ever to do research aboard the International Space Station with the frequency of spaceflight, the frequency of launches, and the accessibility of the implementation partners who are helping us. So for the junior folks in the audience, I think you're really in a golden age of being able to be involved in this sort of work. I was a graduate student when I just got started, but of course there's the Genes in Space program, folks in high school who are getting involved with this sort of work. So it's the perfect time to be involved in this area.
Absolutely. And so when you were looking to do this type of research, how did you even know how to culture something? You maybe reached out to Stephanie here at BioServe that showed you the way, or?
Yeah.
Tell us about that process and how that connection was made.
Yeah, that's an understatement. I mean, BioServe and implementation partners they've had so much experience in doing this sort of research aboard the International Space Station. And I'm a cell biologist, a stem cell biologist, and a heart biologist. I didn't know at the time very much about how to do research aboard the International Space Station and what that entails. I think a lot of it is also being able to speak the same language. So certain things like, what is LEO? What does LEO stand for? What is, so many acronyms that I learned over the process of doing these projects, but also the technical part of it. In our laboratories on the ground, we can grow our cells in cell culture plates. And if we open the dish, we don't have to worry about the nutrients escaping into the air. But that's a very realistic possibility on the International Space Station. And we have to work with folks like BioServe to custom design pieces of hardware that can facilitate the science that we actually do on the space station. And I think that's where your expertise was invaluable. Yeah,
tell us a little bit about that, Stephanie.
Yeah, well, when we go to work with a scientist, I think, the life sciences is our area of expertise. So on our team, we have both engineers and scientists that speak the language of our partners with whom we work. And one of the first things we do is really try to understand what the science is and what Arun's science was and Dr. Wu's science and how they envisioned the experiment operating on station. And one of our areas is really of expertise is to translate how they do it in the ground to being able to successfully complete it in a microgravity environment. As Arun said, you can't just pour liquids, you can't have just open containers, and while you could have an open container where surface tension may hold the liquids in, we also have not only issues around having no gravity, but we also have safety requirements with levels of containment. And so we work really closely with the science team to understand the science so that then we can implement it in hardware that makes sense for the science. You know, the other thing that we do that I think probably Arun realized pretty quickly is a lot of testing in conjunction with the science team. We want the science team to do their testing, but we provide hardware early on so that they can do a lot of testing so we have the best opportunity to have a successful experiment for sure. There's a lot of back and forth, and in that, not only does it give Arun's team a chance to learn about the hardware and you know, and some of the limitations of hardware, because we can't be exactly like it's done on the ground most of the time, but also we can learn some of the intricacies of the science, and it helps us implement his experiment better.
So how does it work? So when we're in this line of research, it has conditions have to be very well controlled. So how does that work with your hardware? How do we ensure, are there astronauts checking the temperature all the time, or live camera feed? How does that work?
So for us, you know, we've had both automated and crew-tended hardware, and so we have incubators on station because we focus in the life sciences. So we provide the temperature control via our incubators, and we can communicate with our incubators from our offices in Boulder, Colorado, and so we can check what temperature is happening in the hardware via those incubators. And then for our crew-tended hardware, when the crew actually pulls that into, say, the life sciences glove box to do a media exchange or to put the piece of hardware onto the microscope to do imaging, we always, for the most part, have an over-the-shoulder video camera so that we can watch in real time what the crew is doing. And we also have our ops lead in our payload operations and command center talking directly to the crew and helping them walk through all of the procedures with the experiment to ensure that they're doing the experiment how we've designed it and as closely as possible to how Arun would do it in his lab. So we have a lot of mechanisms to pay attention to what's happening with the hardware, video to see the crew operating, we take still images, you know, some of our hardware has sensor systems, so it really just depends on the science and what those requirements are.
How would someone get in touch with you? So if I'm, you know, sitting in a lab, I have this great idea, I don't, you know anything about this. How do I get in touch with you? And then how do I know what type of equipment and resources that you have to provide?
Well, I think I do think the ISS National Lab website is a really good resource. They have a list of all of the organizations like us that provide hardware. So obviously they can go to the ISS National Lab website. We're part of the University of Colorado. We have our own website. You could go to our website directly. And you know, and we have a list of our hardware, but I think the best thing to do is to call us, and I usually have a conversation first with all the scientists that contact us about potentially supporting them and finding out what their needs are, and then talking about the different types of hardware that we have available. We have lots of different designs of pieces of hardware that can support a wide variety of life science research in addition to stem cells, and we do a lot of modifications to that hardware. So we have kind of a base platform that can be modified potentially, and then
so
sometimes
we do a specific request and you'd be able to.
Yeah, so it really depends upon the requirements of the science, and then we based our hardware around that, what those requirements are.
Okay, interesting. Maybe to both of you, what do you see the future of this type of research in space? What should we be looking for next?
Yeah, I think on the technical side of things, I think there's a few things that I'm really excited about when it comes to the next phase of life sciences research in space. One is incorporating automation into the work that we do. I think it's great to work with the astronaut partners on the space station, but I do think there's a particular advantage for having automated platforms for cell culture, for example. You can increase the throughput potentially with the experiments that we do. And as we know, reproducibility is something that's critically important for science, not just in biomedical research, but science across the board. And so incorporating these automated approaches, incorporating high throughput approaches, I think will help take our science to the next level.
Okay.
I'm excited about the variety of science that's being conducted on the space station right now, and I'm excited about some of the in-space manufacturing programs that are occurring right now. I think that that's really expanding the types of science that has historically been conducted onto the station. And I think in the future what's going to happen is doing all of that variety of science now is going to help narrow down the types of, in terms of, you know, generating a product, the types of science that really can benefit from a microgravity environment. I think that we should always have the fundamental science happening, I think both for space exploration, but for benefits to human health, and because it really starts at the fundamental science level. But I think that from the in-space manufacturing perspective, that we really are going to, in the future, see certain types of science, and hopefully in the stem cell arena, that is really benefiting from the cells from being expanded in space or grown in space, and then those products will be brought back down to Earth to benefit humans. I also think with the advent of potentially the commercial partners in the commercial space stations will help streamline some of the science that we're able to do. Obviously, this space station is a government platform, and there are a lot of regulations and protocols that we have to follow. And I'm looking forward to potentially with the commercial platforms having a streamline in terms of doing science and expanding the ways that we can do science on those platforms.
And do you envision that as we're moving to the commercial platforms, there'll be opportunities for not only just demonstrating basic fundamental properties of cells in space, but then taking that on to the next step to becoming a product?
Yeah, I mean, I think that's obviously the holy grail, is to be able to develop a product in space that's better than being developed here on the Earth, and that we can, and that it's, you know, commercially viable. I mean, that has to be part of it, and so I really think that's the holy grail. And I think that all of the work that's being done now to support that potential is going to lead to, you know, something in the future, for sure.
And speaking of what's next in the future, I definitely want to mention that as part of this session, so you will all vote this is the most exciting session, we have a launch that's coming up in a couple of hours. So congratulations to you on that.
Thanks.
We're very much looking forward to that. So just one last question. What about the potential for, you know, this far out moonshot thing? What about the potential of developing organs in space?
Yeah, I guess it's a question that you know, draws a lot of attention. I think, and I agree with what you said, Stephanie. I think we have to really focus on the basic science first before we can even kind of really think about making those grand dreams a reality, because right now, we're still trying to understand what happens to these cells in space. How do stem cells divide better? How do they turn into other cell types potentially better in space? And if we're able to definitively prove that, then we can start thinking about those next steps in terms of, okay, let's build a little bit beyond that, make slightly more complex tissues and not even organs, but small tissue constructs that have, say, blood vessels. And if that's beneficial, and if microgravity has a positive influence on that, then really we can take that further step and perhaps develop organs one day. And, you know, it's always nice to dream. I think perhaps 10 to 15 years down the road, we can make it a reality.
Okay. Yeah.
Yeah, and I agree with that. I think, you know, it's definitely small steps. It's obviously very complex. If it was easy, we'd be making full organs already on the Earth, and we're not. And so while the microgravity environment could add some benefit, I think it's, you know, we have a long way to go, both from a science standpoint and a technological standpoint to be able to support that type of work. But the fact that we're supporting the science that would be needed to be completed to get to there, I think is just really super exciting. And you know, I've been doing this for a really long time, and the community has grown just by leaps and bounds, just even over the last decade. And to me, that just provides the opportunity, the ability for innovation and for, you know, the next generation to think about these types of questions and to figure out the way that we will get that done in 10 or 15 or 20 years. So.
Mm-hmm. Speaking of growth of the community, it's not just NASA that's doing these type of studies, and I understand that you participated, your research was flown on not just a NASA mission, but also on a private astronaut mission. How was that for you?
It was a tremendous experience. I think, you know, it was in large part equivalent to what we have experienced in the past, and, you know, we had such an excellent time working with our partners, not only at BioServe, but also Axiom Space. We flew aboard the AX-2 mission not too long ago. And, you know, we were working with the Axiom astronauts who did our hands-on work for us aboard the International Space Station, and we were so excited about what we saw. We actually have a manuscript currently in revision, under revision right now, and hopefully will be published pretty soon about some of our findings. But it was a tremendous experience to be a part of. I think it was, you know, a milestone moment, I think, really.
And I just add to that, the exciting thing about that, exciting and challenging, was the timeline. It was very rapid compared to what we typically do, and the length of time that it takes. And so I think one of the reasons why it was so successful as well was because Arun's team was really great at doing the science. Like, we'd say, hey, we think you need to test, you know, A, B, and C, and his team just went and did it. And so that nice collaboration between all of the partners really is really what made his project successful. Mm-hmm. It's like, you know, Axiom had the platform, we helped, you know, support his experiment, he did the science, and that collaboration really made the difference, that successful collaboration. It could have been very difficult just because it's a scenario that we haven't been doing, but it was really successful, and it takes, you know, motivated teams like we had to get that done successfully. So kudos to you and your team, Arun, for sure.
Yeah, and definitely this relationship working together seems to really play a major role in the success of these missions. And one of the things you touched on was this rapid turnaround. And I think as investigators, that's one thing that we're really looking for, is the opportunity to fly multiple missions with rapid turnaround. And perhaps this, you know, I'd like your opinion, but perhaps this is the way of the future. You know, I know we made this announcement today about, you know, inspirational ideas. So do you think that this is really going to be the way of the future? And, you know, we should ensure people out here who might be interested in entering this field that they're going to get rapid turnaround in multiple flights?
Yeah, I think that's really an important component in discovery in microgravity research right now. You know, historically it's been one-offs in the science. You worked for years, you got one science experiment, you had one chance at it, you got it done, and then there was another scientist that had a different project, and which was great because it really laid the basis of a wide variety of different types of research. But having the opportunity to do your science, multiple experiments in a short period of time, I think will really help push the field even further than it is right now. And, you know, because in the lab, you think about it, you do one experiment, you know, in a couple of weeks, and you do another one, and you do another one, and, you know, in a year's time, I don't know how many experiments you do in your lab, but certainly more than one. And so having the opportunity to do multiple experiments, and still not as rapidly as you would do in your lab, it's just logistically not possible, but having that opportunity to find, get results, make some, you know, decisions on what your results indicate, and then being able to do a follow-on experiment, I think will really help push the field even faster.
Great, thank you. I think we're kind of running short on time here. Do we have some questions from the audience?
Yes, so we have time for just one question. And I guess this would go for both of our speakers. You both talked about automation at various times. Is there benefits to having the crew involvement in the experiments?
Sure, absolutely. I think one thing that's very beneficial is potentially changing an experiment on the fly. If there's an observation that's made, and this is actually something that happened to our experiment aboard the AX-2 mission, if there's an observation made about how cells are growing, for example, and we need to adjust our experimental parameters, I think having crew on hand to make those changes is extremely beneficial. That's not something that we can perhaps as easily do with automated platforms right now. Maybe in the future it'd be more possible, but certainly there are huge advantages to having crew working hands-on in space.
Yeah, and I would just add to that, I think, you know, there's pros and cons of both an automated system and a crew-tended system, and definitely the flexibility that you get with a crew-tended system. And particularly now in the early stages of some of the science that we're supporting, doing a crew-tended experiment allows you to do things that you really cannot do in an automated system right now. There are lots of things that you can do, but there's also things that you can't do. And so having the ability to do both really, I think, again, pushes the science to a level that if you just had one or the other, that you wouldn't be able to do.
Okay. Any other question, or are we out of time?
Yeah, I think we're out of time, but we really enjoyed the conversation.
All right, yeah, and I just, just want to echo one last thing that Arun said, which is really we are in the golden age of doing this type of research, so I really encourage you while you're here to take the advantage while the investigators and the flight implementation partners are here to start up those conversations, and we look forward to all the exciting research that's going to be done. Thank you.